In biological engineering, one of the most significant challenges is optimizing a system where the number of possible combinations is, for all practical purposes, infinite. Progress in this area is often constrained by fundamental limits of labor, time, and cost. Today, we're going to examine a workflow that has been developed to address this very problem.
So, here's our plan. We'll start by outlining the core optimization problem in biology. Then, we will introduce METIS, which is an active learning workflow. We'll examine its performance across 3 distinct applications. First, a gene circuit optimization. Second, a transcription and translation unit. And third, a synthetic CO2 fixation cycle. Finally, we'll consider the implications of this for the future of biological systems design.
You know, to effectively engineer biological systems, we need a combination of efficient experimental approaches and powerful computational methods. Machine learning is an obvious candidate, but its application is often hindered by 2 primary obstacles: the requirement for deep informatics expertise and the need for large, manually labeled datasets, which are both expensive and time-consuming to generate. To work around the constraints of limited experimental capacity, researchers can employ a strategy known as active learning, or Bayesian optimization. Now, instead of proceeding based on intuition or brute force, this approach allows the model to guide the experimental process. It learns from existing results and then suggests the next set of experiments that are most likely to yield improved performance, thereby saving considerable resources. And this brings us to METIS. METIS stands for Machine Learning Guided Experimental Trials for Improvement of Systems. It's a workflow developed to make active learning more accessible. It was built with experimental scientists in mind, those who may not have a background in programming, and it gives them a structured methodology for using data to drive their research forward.
The workflow itself is a straightforward, iterative cycle. It begins with the user defining their objective function and the relevant factors, or variables. METIS then generates an initial set of experimental combinations. After the wet lab experiments are performed, the results are input back into the system. At this stage, METIS analyzes the data using an algorithm called XGBoost, which is particularly effective for learning from smaller datasets. Based on this analysis, it then suggests a new, more informed set of experiments for the next round.
Okay, so let's examine METIS in a practical application. Our first case study is the optimization of a gene circuit, specifically a LAC-I-based multi-level controller. The objective here was twofold: to maximize the fold change, which is a measure of the signal-to-noise ratio, and also the dynamic range, which quantifies the total protein output.
After 10 rounds of METIS-guided experiments, the combined objective function showed a moderate improvement. However, a closer look at the metrics revealed a problem. The fold change, that critical signal-to-noise metric, had shown no significant increase. This suggested that there was an underlying issue with the system that could not be resolved by simply adjusting the component concentrations. And this is where a key function of METIS becomes particularly useful: its feature importance module. This tool identifies which factors are exerting the most influence on the system's output. The analysis revealed an unexpected contributor: the concentration of the PT7 LAC-I plasmid. This component was intended only to supply a repressor protein, but it was apparently dominating the system's performance, indicating it was the likely source of a bottleneck.
So this disproportionate influence from the LAC-I expression plasmid required an explanation. The data implied that its effect was in fact detrimental, which led the research team to formulate a new hypothesis.
The hypothesis was resource competition. You see, both the repressor plasmid and the GFP reporter plasmid were utilizing the same strong T7 promoter. This meant they were competing for the same limited pool of transcriptional and translational resources, like T7 RNA polymerase and ribosomes. This competition was suppressing the production of the reporter protein, which accounted for the low fold change.
To resolve this bottleneck, the team replaced the LAC-I expressing plasmid with purified LAC-I protein, thereby eliminating the resource competition entirely. The effect was substantial. After just a single additional round of optimization on this revised system, the fold change increased to 123. This demonstrates how the workflow can not only optimize a system, but also help diagnose its underlying architectural flaws.
The second case study demonstrates the ability of METIS to handle categorical factors, in this case, different DNA sequences, as opposed to purely numerical values. The objective was to optimize a transcription and translation unit that was composed of a library of discrete sequence variants.
The system was constructed from variants of a promoter, a ribosome binding site, and protein end sequences,
This created a combinatorial space of 10,800 potential conditions. Using METIS, the team was able to explore this space by testing just 200 combinations over 4 rounds in a cell-free system.
The cell-free optimization was successful, increasing protein yield by up to 12-fold. However, when the top-performing combinations were tested in vivo, that is, in living cells, the performance did not translate well, showing a low Pearson correlation of 0.14. This is an important finding as it highlights the common disparity between in vitro and in vivo results. But by feeding this new in vivo data back into METIS for just one additional round, the researchers improved in vivo performance by 130%.
The final application tests METIS on a particularly complex system, the CET-SEACH cycle. This is a synthetic in vitro metabolic network for CO2 fixation, and it involves 27 different variable components, including 17 enzymes and 10 cofactors.
The number of potential combinations in this CETH cycle is quite large. With 27 variables, the theoretical search space is on the order of 10 to the 25th power. A number of this magnitude is simply impossible to explore using traditional experimental methods.
Despite this complexity, METIS was able to navigate this landscape using only 1,000 experiments conducted across 8 rounds. This highlights the efficiency of this data-driven optimization approach.
The results were quite significant. In the first phase, the team optimized for productivity, and METIS identified conditions that increased the yield of the final product by more than 10-fold. Then, they shifted the objective function to efficiency, that is, yield per unit of enzyme. This second optimization phase resulted in a nearly 6-fold improvement in efficiency, creating what is, to date, the most efficient in vitro CO2 fixing system described.
Once again, the feature importance analysis provided critical insights. It identified the enzyme MC-ACO as a key factor for productivity. For efficiency, it highlighted HPBS and coenzyme B12. Interestingly, the analysis also uncovered a non-intuitive relationship. For both HPBS and B12, higher concentrations were found to be detrimental to the cycle's performance.
So, what is the broader significance of a tool like METIS? Well, it represents a step toward making powerful machine learning tools accessible to the wider community of biological engineers and scientists directly at the lab bench.
The authors of the study summarize this well, stating that this workflow can help to standardize and accelerate the classic design, build, test, learn cycle. This makes the engineering of complex biological systems a more systematic and efficient process.
The essential takeaway here is that METIS offers scientists an accessible workflow to optimize highly complex systems with substantially less experimental effort. Furthermore, it functions as a diagnostic tool, capable of revealing previously unknown system interactions and performance bottlenecks during the process.
And this brings us to a final forward-looking question. As these data-driven, active learning approaches become more widely adopted, what complex biological systems, from novel metabolic pathways to entirely synthetic genetic circuits, will be the next to be engineered and understood?